It's my pleasure to be back in Boston today to be able to present high-end analytical tools to meet the latest regulatory requirements for generic peptides. First of all, a few words of introduction about the PolyPeptide group. PolyPeptide has more than 70 years experience in peptide manufacturing with 1,200 employees worldwide. We have a network of 6 CGMP certified sites on 3 continents, 2 on the west coast of the USA, 3 in Europe, Sweden, Belgium, and France, and 1 in India. Also headquarters in Switzerland. We are a global market leader for innovative clinical and commercial stage peptide drug substances. We are manufacturing around half of all approved peptide APIs in the world, and we are a long-term partner for customers from biotech startups to large pharmaceutical companies. So on the agenda today, I realize we have lots of acronyms. So we will be talking about UPLC, HRMS, 2D LCMS, MSMS, peptide mapping, and finally LCMS for peptide quantitation. So let's start with UPLC for impurities quantitation. Ultra-high performance liquid chromatography has become state-of-the-art over the last 20 years. On this chart, you see the evolution of this liquid chromatography technique from 10-micron particles back in the 70s down to sub-2 micron particles from 2004 when Waters started this technology. At the same time, you see the increasing resolution power of those sub-2 micron particles. The benefits are higher peak capacity and resolution, higher sensitivity with less peak broadening, and shorter analysis time with higher flow rates. Obviously, there is a drawback coming with that. It's the high back pressure, typically from 600 to 1,000 bars. So HPLC today is probably a must to meet the increasing regulatory requirements, which have previously been described by Jamie. Here again, we have a copy of the guideline, most recent guideline from FDA for ANDAs for certain highly purified synthetic peptide drug products that refer to listed drugs of recombinant origin. In this guideline, no single impurity can be above 0.5%, and any impurity above 0.09% must be identified, meaning it must be specified upfront, and that's the real challenge. So in our experience, the major considerations for UHPLC methods development are the LOQ, which should be around 0.05% to meet the target on the unspecified impurity limit. Primary methods, which should preferably be LCMS compatible in order to properly identify impurities. One or several orthogonal methods should be developed for potentially coeluting impurities, including the potential isomers, and we'll talk about that later. And finally, in order to properly identify the impurities on a routine basis, an identification standard should be implemented both in IPC, QC for release and for stability studies. So here is an example of a primary UHPLC method for quantitation of impurities that we have implemented for a very recent generic peptide. It's using a Waters Acuity CSH C18 column at 30 degrees with a 0.1% TFA acetonitrile gradient. Therefore, it is LCMS compatible. We have an LOQ of 0.07%. And we are spiking known impurities, in order to properly identify every single impurity without any ambiguity. So we are showing here a couple of API batches that we have manufactured together with the identification standard. Here is routine batch release data for this generic API. The robust analytical method that we have developed allows consistent impurity profile determination. You see consistent purity and even increasing overall purity over time over those 12 batches. And you see consistent purity data over the impurity data over for the different impurities. On the last line here, you also have the high molecular weight product results. We're using size exclusion HPLC for this impurity. You see that consistently the high molecular weight product is below 0.02% or up to 0.03%. We also use this primary HPLC method for comparability with the RLD. Here, if we compare the purity of our synthetic API with the RLD, which is of recombinant origin, we have above 99.0% purity for the API against 97.5% for the RLD. A few of the impurities are common between the two processes, but most of them are different naturally. And again, there is no impurity above 0.5% in our API, and any unspecified impurity is below 0.10%. Now, the challenge of isomeric impurities. Because isomers cannot be detected by LC-MS when they are coeluting with the main peak, we need to perform a systematic preparation and spiking of all potential isomeric impurities. In this case here, we have evaluated 28 isomers, so all potential D-isomers for the given peptide, and the beta-asp analogs, which also have the same mass and are not detected by LC-MS if they are coeluting. On the right here, you have the relative retention time for all potential isomers. And as a matter of fact, you can see that 90% of the isomers are separated in this case. So there are 10% remaining that needs to be addressed by orthogonal methods. This is shown on the slide here, specifically for the two isomers, the D amino acid 6 and D amino acid 7 in the sequence, where we have developed an orthogonal method. It's using a water acuity BEH C4 column at 30 degrees and a mobile phase made of ammonium phosphate at pH 3.6. The data shown here indicates that those two potential isomers are actually not present in our API batches. Similar approaches have been conducted for the other isomers that are not separated by the primary HPLC method. Now, moving on to high-resolution MS and LC-MS for impurities identification. We start talking about high-resolution mass spectrometry when we get a resolution above 20,000. As you probably know, typically peptides ionize as several multiply charged ions. So here you have a peptide of 6,153 Dalton, and you can see the different multiply charged species, doubly, triply, quarterly, and 5-time charged ions. The beauty of HRMS is that it gives access to detailed impurity profile, isotopic profile for each observed ion, which makes the charge state determination very easy. So if you look at the first isotopic profile here, you have a mass difference between the different isotopes of 0.33, which means it's a triply charged ion. Similarly, on the second isotopic profile here, you see a mass difference of 0.5 Dalton, meaning that this is a doubly charged ion. And once you have the charge of the ions, you can very easily go back to the monoisotopic mass. So here, HRMS allows very easy charge state determination and very easy monoisotopic mass determination. In addition to that, due to the progress of bioinformatics, it's now possible to automatically deconvolute MS spectra, which also allows very easy results interpretation. So on the top of the chart here, you have a raw MS spectrum with the different isotopic profiles that you see on both sides. It can be automatically deconvoluted, meaning conversion of all multiply charged ion into a singly charged ion here. And then where the monoisotopic profile is preserved, as you can see. And then this monoisotopic profile can be deisotoped into a single monoisotopic mass. So we go from a relatively complex raw MS spectrum to a very easy monoisotopic mass determination. The mass difference between impurities and the expected structure is the basis for impurities identification. Here we have a number of examples of mass differences for different amino acid residues, for different protecting groups, and for typical modifications, and some of them we've discussed earlier, such as deamidation here. You will see that in some cases, just having the difference in mass doesn't allow direct identification of the impurity. For instance, if you have a plus 71 mass on your impurity, it can be alanine, can be, well, sarcosine if you have sarcosine in your molecule, but it can also be ACM if you have an amino acid protected by ACM, and it can also be a beta-ala insertion. So it's much more complex than just relying on the mass, but the mass determination is really the first step in the process. Very often, additional characterization is required to identify the position of the modification. Obviously, as I already mentioned earlier, modifications such as conversion of ASP into beta-ASP or racemization of some amino acids cannot be detected by LC-MS if those impurities are coeluting with the main peak. Here is an example of a stress study where we have used LC-MS for the determination of the degradation products of our generic peptide. So we are using LC-UV for quantitation of the impurities, and we are using LC-MS for identification of those impurities. We are running exactly the same analytical method in HPLC-UV and HPLC-MS. So here you see the unstressed material, the high purity generic peptide, and then you see basic, oxidative, acidic, heat, and UV light impurity profiles or degradation products. With LC-MS, we can detect the mass difference between the impurity and the main peak, and therefore we can start assigning identification of the impurities. For instance, here we don't have any difference, so these are isomers. Plus 16 is an oxidation, which is expected in oxidative conditions. In acidic condition, we have oxidation as well, double oxidation, and acetylation. And by the way, we also see a deamidation here, which we discussed earlier. And with temperature, we see imine formation, which is quite common when histidine is present in the molecule. And also, we see a dimer peak here, which is eluting in the reversed phase chromatography. So LC-MS is really a very important tool to identify your impurity profile both at batch release and also during stability studies, or stress studies, at least. LCMS is also used for peak purity confirmation, as we discussed in the previous talk. So in order to perform this peak purity confirmation, we screen the main peak, we screen the MS spectra across the main chromatographic peak. So here we have the beginning of the chromatographic peak, the middle of the chromatographic peak, and the end of the chromatographic peak. We perform the deconvolution and de-isotoping, which I explained earlier, and then you have your simplified spectra across the chromatographic peak. In this case, where it is a non-stressed sample, we see that there is no coeluting impurity along the chromatographic peak. Now, if I move to the temperature-stressed sample, it's the same example as before. When we do this screening of MS spectra across the chromatographic peak, we start seeing at the backside of the main peak the start of the imine impurity, which is eluting. So we see the M+12 becoming present at the backside of the main peak here. Sorry. LCMS can also be used when running size exclusion HPLC, as long as the size exclusion HPLC is compatible with LCMS, which is the case here. So the high molecular weight product method is using Waters BEH SEC 125 column and an isocratic water acetonitrile IPA TFA system. We have the baseline here with a little bit of carryover. We have 3 batches of API, and we have implemented a control. We talked about the control at the end of the last talk. We have implemented here a control where we actually generate a high molecular weight product impurity by submitting the API to 80 degrees temperature in the solid state overnight. This way we have a control of the high molecular weight product retention time. And actually, when running the LCMS with this method, we can identify the high molecular weight product impurity peak as a covalent dimer with a mass of 8.2 kilodalton, whereas the main API peak is at 4.1. So it's really a covalent dimer here, probably resulting from cross-linking of side chains in the peptide molecule. Now, moving to 2D LCMS, which is used for impurities identification as well. However, this technique is particularly useful when you're running non-LCMS compatible chromatographic methods. For instance, when you're using TAP, phosphoric acid, perchlorate, sulfate methods that are not LCMS compatible. So when running 2D LCMS, the principle is to perform a first dimension LC run with the non-LCMS conditions. This is the case here with the phosphoric acid HPLC mobile phase. And then running a second dimension LC with LCMS compatible conditions, such as formic acid or TFA, for example. It's a very short chromatographic run. The impurity peak, which has been collected in the first dimension, is injected, and the salts are removed, and the impurity peak is analyzed by LCMS. This way, we get direct access on to the mass of the impurity in this second dimension. Here, by performing the hard cutting of the tailing impurity here, we identify the impurity as an endoglycine analog. A few words about the equipment. In this case, we're using an Ultimate 3000 UHPLC system coupled to an LTQ Orbitrap XL system. The main difference with the regular HPLC system is that here we have two pumps. We have a low pressure quaternary pump for the first dimension liquid chromatography, and then we have a high pressure binary pump for the second dimension liquid chromatography. In between the HPLC system and the mass spectrometer here, we have a connecting valve with 6 ports and 2 positions. Initially, the loop is in standby when we are running the first dimension chromatography. Then we load the impurity into the loop here at the time of hard cutting the impurity. And finally, the valve is switching into the position 1,6 in order to inject the collected impurity into the second dimension LC. I will show 2 examples where we have used this 2D LC technique. Here, in this first example, we're using a method from Pharmacopoeia. It's a sulfate method, so it's not LC-MS compatible. We cut the impurity A here, and we inject it in the second dimension using formic acid, which is LC-MS compatible. And in this case, we can detect this impurity as a Des-Ala impurity. You will see that we also see some sulfate adducts coming from the mobile phase of the first dimension liquid chromatography. In the second example, we are even identifying several co-eluting impurities in a single impurity peak. So beforehand, we knew that the peak here was mainly a DASN peak, but by hard cutting this peak at the beginning, in the middle, and at the end, we can see that it doesn't only contain the DSN impurity, but also Des-Asp impurity at the beginning of the peak, and also the Des-ASN impurity towards the end of the peak. So it's really a very powerful technique to closely identify impurity profiles when you're using non-LC-MS compatible mobile phases. Now I will switch gear a little bit and talk about MS-MS for peptide sequencing. And first of all, peptide sequencing is performed by electrospray MS-MS. We perform fragmentation along the peptide backbone, and we generate B and Y ions. So B ions are from the N terminus, and Y ions are counted from the C terminus of the peptide molecule. The sample is infused into the ion source directly, so it's not an LC technique, it's a direct infusion of the sample. The ion from the peptide to be sequenced is selected. It can be doubly, triply charged. And then fragmentation is performed by high collision dissociation and or by collision-induced dissociation. These are complementary fragmentation modes. This way we can generate B and Y ions, and we can compare them to theoretical fragments to perform the peptide sequencing. And the full peptide sequencing is confirmed if the complete sequence has been covered. This is what we call a top-down approach, as we are starting from the peptide. Here on the chart, you can see at the top the raw MS-MS spectrum. And again, by deconvolution and de-isotoping using high-resolution MS, we can simplify this complex MS-MS spectrum into a more easily interpretable spectrum, where we can recognize some of the B and the Y ions of our peptide. Then it's a matter of writing down the sequence of the peptide here. and calculating the mass, the theoretical mass of the B and Y ions, and then when we compare the observed to the theoretical B and Y ions, we can confirm full sequence coverage of the peptide. So you see here, only with the B ions, basically, when we use those two fragmentation modes, we can cover the full sequence of the peptide. Now, I would like to talk about peptide mapping and LCMS technique for impurities identification. So, peptide mapping gives access to specific peptide fingerprints. It's performed either by enzymatic or by chemical cleavage of the peptide sequence at specific amino acids. So if we take, for example, enzymatic digestion, if you take trypsin here, it's cutting the peptide at the C-terminus of arginine or lysine, so basic residues. Similarly, chymotrypsin will cut at C-terminus of tryptophan, tyrosine, phenylalanine, leucine, MET, and ALA. So we have a whole toolbox of enzymatic agent that can be used to conduct those peptide mapping. Peptide mapping can be used in two ways. First of all, for identification of long peptides against reference standard by LC-UV using the characteristic fingerprints. And secondly, through the simplified impurity profile specific to each generated fragment, it allows impurities identification through LCMS and LCMSMS of the peptide digest, which I will show in a minute. So here in the first example, it's a simple identification of our peptide API by LC-UV comparison with the reference standard. So we have the reference standard in blue, which gives typical fragments. So you see the 1,3 fragment, the 4,9, the 10,15, the 16,21, and the 22,31. And when you run your peptide sample, you see exactly the same fragments in pink, confirming that the peptide sample is the same as the reference standard. Now, for identification of impurities, we need to use LCMS and LCMS-MS. So in this case, again, you recognize the main fragments, which I just described. And first of all, we try to identify where the modification plus 106 is located in the peptide sequence. So we look for all potential plus 106 modifications on the different fragments. And as a matter of fact, this modification is only found on fragment 2231, which means that this is where the modification is made. In a second step, we are performing LCMSMS, so online sequencing of both the original fragment, 2231, and the modified fragment. So from the unmodified fragment, we recognize we observe a few of the theoretical B and Y ions, as previously. And now, on the modified fragment, we can confirm that the modification is positioned at amino acid 25 because we find the corresponding ions with the modification on amino acid 25. And then, starting amino acid 26, we find the same masses as previously. So this is a very powerful technique to identify the position of modifications, and it's complementary to the initial LCMS testing that is performed. And finally, I would like to talk about a technique that we have implemented recently on site, which is LCMS for the actual quantitation of impurities. So in this example here, you will see that we actually quantitate 10 different impurities in a single chromatographic run. It's a relatively short method, and as a matter of fact, a number of impurities are not separated one from the other. In order to perform this quantitation, this simultaneous quantitation of 10 peptide impurities, we use a single quadruple MS equipment, which is a low-resolution equipment. We perform the quantitation against internal impurity standards using single ion monitoring detection mode. The acquisition rate is 15 points per second for each compound, and we performed automatic peak integration with full data integrity. In this specific case here, we preferred using doubly charged ions because they form less adducts than singly charged ions. So you have the doubly charged ion here and the singly charged ion here. So the charts here show the SIM MS traces, so the single ion monitoring traces, and sorry. And here you have three charts with the impurities spiked at 1%, 0.5%, and 0.1%. This method has been validated for routine QC release testing. So we have validated linearity, LOD, LOQ, accuracy, repeatability, intermediate precision, and even robustness. And very interestingly, you can see that LOQ is very close to the 0.1% that we discussed earlier. This is batch data over 17 batches of this peptide release. And again, we get very consistent data for the different impurities over time. Manufacturing process is very robust. All right, so as a summary, I hope I was able to show you our analytical toolbox for peptide, API, and impurities. A number of techniques have been described for API characterization, quantitation, and sameness with RLD, as well as impurities identification, quantitation, and characterization. So in conclusion, as we have previously discussed, regulatory requirements are increasing for peptide APIs, not only for generics, but also for NCEs. And specifically, there should be no unspecified impurity above 0.09% for generic APIs and for complex peptide APIs. Even though the analytical tools have greatly improved to identify and quantitate impurities in peptide APIs, supply chain, process development, and manufacturing expertise are still required to consistently deliver the expected peptide quality and avoid generic peptide storage. With that, I would like to thank my colleagues from PolyPeptide, David Kosker and Emmanuel Gay, our MS specialists, Pauline Lacaze, Cédric Delannoy, Didier Monet from Analytical Development. And I would like also to thank you for your kind attention.